Click the bell icon and subscribe to our channel so that you will not miss any classes.
Hello students, welcome to the class. Now we shall discuss the last phase in the mitotic cell division, that is the telophase. The telophase begins with the pole movement of the separated chromosomes completes. Separated chromosomes will complete. This marks the beginning of telophase. That means in the anaphase we have discussed that the chromosomes have started moving towards the opposite pole like this. They started moving towards the opposite pole. This movement has been complete. And there is a formation of two groups of chromosomes. There is a formation of two groups. One group here and another group here. So there is formation of two groups of chromosome. This marks the telophase. So you should understand that the telophase, when it starts, in the beginning itself I told clearly, we cannot draw a strict line between the phases, but we can study the beginning of the phase and the end of the phase by some events. As like the telophase starts when there is a formation of two groups of the chromosomes, and in the two groups of chromosomes the chromatids starts decondensation. Whenever you remember from the prophase that the condensation of chromosome will happen in the prophase, the opposite events will happen in the telophase, which means in prophase the condensation of chromosome will happen. In telophase there is a decondensation. Whatever the chromosome has been formed, chromatids has been formed, they will lose their integrity and they undergo the process called decondensation. As a result, what happens? As a result, the chromosomes again, they appear like a thread, like this. They again appear like a threads, and there is a formation of nuclear membrane around the disintegrated chromatin network. There is a formation of nuclear membrane. So formation of nuclear membrane. In prophase there is a disintegration of nuclear membrane, and there is a appearance of the nucleolus. The nucleolus will start appearing in both. So that means whenever you understand that the opposite events will happen, there is a decondensation of the nuclear material or the chromosomes. There is a formation of nuclear membrane around the two sets of chromosome, and there is a formation of nucleolus. As well, the other cell organelles like Golgi apparatus, endoplasmic reticulum, all of them start appearing. Whatever they disappeared in the prophase, they start appearing in the telophase. By the end of telophase, complete structures will be formed in the two sets. So that is the telophase. But remember one thing, there is a slight presence of the spindle fibers. Spindle fibers present for some time. They will not disappear completely as such. They take some time to disappear. So here they present for some time. By the end of telophase, they also get disappear. So these are the events during the telophase. In the telophase, the completion of poleward movement of the chromosome, separated sets of chromosome will happen, and after reaching the complete poles, the chromosomes will undergo or they lose their integrity and they undergo decondensation. As a result, there is again formation of chromatin network. There is a formation of nuclear membrane around the two sets of chromosomes. There is a formation of nucleolus and the other cell organelles like endoplasmic reticulum, Golgi apparatus. But the spindle fibers are present for some time. By the end of telophase, they also get disappeared completely, which completes the division of the nucleus, that is karyokinesis. So if you recall from the beginning, karyokinesis will happen in 4 phases: prophase, metaphase, anaphase and the telophase. Now the telophase is complete. It will lead to the next step in the mitotic division, that is the cytokinesis. The mitosis will not happens in one step, that is the division of nucleus. It will happens in 2 stages. One is karyokinesis followed by the cytokinesis, which means the division of cytoplasm. Whenever there is a division of nucleus, to maintain the ratio of nucleus and the cytoplasm and to maintain the cell size and to maintain the cytoplasmic content in a cell, the cell has to divide its cytoplasm immediately after completing the division of nucleus, which is called as cytokinesis. Cytokinesis is the division of cytoplasm. And the division of cytoplasm is different in plant cells and animal cells. Now we are discussing with the animal cells. So we shall take up first the cytokinesis, which is happening in the animal cells. In animal cells, the cytokinesis will take place by the formation of furrow in cell membrane. Furrow in cell membrane, that is after the formation or after the complete division of nucleus, the cell will appear just like there are 2 nucleus in a cell after completing the telophase. So this is one and this is another nucleus. It just appears like that. To begin with the cytokinesis, here from both the sides of the plasma membrane, there is a formation of this furrow. This is the furrow. The furrow will be formed from both the sides and the furrow will get deepens. The furrow will get deepens like this. The furrow will get deepens like this. Here one nucleus and here one more nucleus. So the furrow will get deepens. Finally the furrow will reaches or they meet with one another, which results in the complete division of the cell, which results in the formation of 2 daughter cells. This is one daughter cell and this is another daughter cell. This will complete the mitotic cell division. So that is the simple thing what happens in the animal cell. In the animal cell, the cytokinesis is very simple by the formation of a furrow in the cell membrane. initially the furrow will form from the both the sides and it will get deepens and join finally, which results in the formation of two daughter cells. Whereas in plant cells, in case of plant cells, since the plant cells cell wall is very thick and it is not flexible. In animal cells, the cell wall is not present. Only cell membrane is present and it is flexible. But in plant cells, there is a presence of cell wall which is made up of a very thick materials like cellulose, pectin. So, here is the animal cell. Because of the presence of cell wall, they cannot form a furrow. So, here is one nucleus. That means it has completed its karyokinesis. So, here the division of cytoplasm will take place by the formation of cell plate. During the division of cytoplasm, initially there is a formation of, there is a beginning of the formation of a structure called the cell plate. So here at the middle there is a formation of the cell plate. Initially cell plate will form initially in the middle, that is between the two nucleus, at exactly at the middle of the cell there is a formation of cell plate. To say technically, the cell plate represents the middle lamella between the adjacent cells. That is, in case of plants, between the two cells, cell wall, here is one cell and here is another cell. Between them, there is a presence of structure called the middle lamella. So, initially, the cell plate formation, when it begins, it represents the cell plate, or we can call it as a cell plate. And this cell plate will grow on both the sides. The cell plate will grow on both the sides like this. The cell plate will grow on both the sides. There is one nucleus. There is another nucleus. And the cell plate will grow on both the sides and reaches the two lateral walls. The lateral walls are already present on both the sides. When it reaches the two lateral walls on both the sides, then it will split the cell into two. So that is, the two daughter cells are being formed. Along with the division of cytoplasm, there is an equal distribution of the cell organelles which have multiplied in the interphase. I told when the cell starts the division, just it has completed its interphase where all the cell organelles are getting multiplied. So multiplied cell organelles are distributed equally. There is a distribution of cell organelles to both daughter cells. It may be plant cell or it may be animal cells. There is an equal distribution of all the cell organelles including the nucleus to both the daughter cells to maintain the stability or to carry out the same functions. So that's about the division by mitosis. So this will ends up with the discussion about the mitosis, cytokinesis as well as the karyokinesis. Now we shall discuss what are the differences between the division, mitotic division in plant cells and in animal cells. Initially, when I started with the mitosis, I told even though the processes found to be similar in plant cells and animal cells, there are few considerable differences between the mitotic division in plant cells and animal cells. We should take up them in detail now. So I am making two poems here in animal cells and in plant cells. In case of animal cells, there is a formation of spindle fibers from the centrioles. From the centrioles, there arises a spindle fibers which are responsible for the separation of chromosomes. Whereas in plant cells, the spindle fibers are not exactly present, but there are spindle fibers which are formed from the nucleoplasm. They are formed from the nucleoplasm. They are not formed from the centrioles. That's why we not call them exactly the spindle fibers. The fibers responsible for the separation of the chromosomes will arise from the structures called nucleoplasm. And during metaphase, in case of plant cells, in case of animal cells, during metaphase, I told in the metaphase there is a formation of cell plate where the chromosomes will arise at the equatorial like this, right? So here the arrangement of chromosomes is very peculiar in case of animal cells. That is, the large chromosomes, that means all the chromosomes are not of the same size, that you should remember. They are of having various size. So this will be visible, or this is possible to study in the karyotype. So that means karyotyping is a process to arrange the chromosomes on the basis of their size and shape, which suggests that the chromosomes are, all of the chromosomes are not of the same size. They are of the variable sizes. So we will be discussing that in the respective topics. So as for now, depending upon the size, the larger chromosomes are arranged at the end, that is here, and the smaller chromosomes are present in the middle. Large chromosomes are present in the middle, in the end. Small chromosomes are present middle. So at both the ends there is presence of larger chromosome and the smaller chromosomes are present in the middle. So that is in case of animal cells. Whereas in plant cells, the chromosomes are arranged. Irregularly in metaphase plate. So there is no such thing that larger chromosomes, smaller chromosomes, but irrespective of their size, they are arranged in an irregular pattern. In case of animal cells, the cytokinesis, as we discussed just now, cytokinesis happens by furrowing. Cytokinesis will happen by furrowing, whereas in case of plant cells, the cytokinesis will happen by the formation of cell plate, which represents the middle lamella between the two cells. So these are the differences between the mitosis in plant cell and mitosis in animal cells. With this, we shall take up two more terms which found to be important for the competitive exams. That is the first one. I will be discussing some terminologies related with the chapter, so that it will be important for studying for the competitive exams like CET and NEET. So those are found to be necessary, I will be discussing. So whenever you have some terminologies which is left undiscussed, you can comment, and I will be discussing that in the upcoming classes. And one such terminology is somatic pairing. Somatic pairing. So this is the special terminology where we have discussed from the interphase that in interphase there is a replication of DNA. There is a replication of DNA which takes place. That means here is the one chromatin network, say considered, and this will get produce its same copy, right? So this we call it as replication. So we write it with a different color. So this is one DNA strand and it produces its same copy. Don't think I am writing in a different color means it's different DNA. The same copy of the DNA. So this should get separated in the interphase. After dividing, it should get separated. But in some cells, the replicated DNA strands replicated DNA strands remain unseparated. They will not get separated, they present together and togetherly they will be carried to the daughter cells after division. So such process is called as somatic pairing. That is, during interphase, the chromatin networks are get replicated. If the replicated chromatin networks are not separated in the interphase, then the such process is called as somatic pairing. Endomitosis. Endomitosis means we have studied from the interphase that the DNA or the chromatids will divide once or they replicate once and produces or double its DNA content. But in some organisms, what happens in interphase, the chromatids divide multiple times. The chromatids will divide multiple times. Instead of once, they start dividing 10 times. That means the same type of chromatids are formed in a more amount. So they get accommodated at one place and results in the formation of polytene chromosomes. That means the repeated division of chromatids in the interphase results in the formation of a polytene chromosome. Such process is called endomitosis. So two terminologies you should remember. So that's all about the mitosis and the difference between the animal cell and plant cell mitosis. So we shall continue with the significance of mitosis and the next type of division, that is meiosis, from the next class.